Now we will start, and I welcome you to the 2020 TEIN Summer Workshop. And for this session, we have prepared a topic of radiopharmaceutical chemistry from clinical needs of theranostics. And as you can see, this trend in the Google trend, you can find that there is an emerging interest for the theranostics. So we have actually extracted, as you can see, the words from the journals of Journal of Nuclear Medicine from 2010 and 2011. And as you can see, the most frequent word that were extracted is actually, as you will expect, PET, and some are FDG and tumors. But interestingly, 8 years have passed, and when we extracted again what are the frequent words from the Journal of Nuclear Medicine, you can see that although the PET and cancers are the main, but there are some words that attract our vision, which are like therapy and treatment, or prostate, or PSMA. So these radiotherapies are gaining
예, 호스트에서 전체 음소거를 시키면 됩니다.
Dok udah mulai yang itu. Endra ya.
Sorry for the delay. So as you can see, what are the increasing high frequency word recently are in this order, which are PSMA and PRRT or DOTATATE, which are related to the therapies. So there are increasing interest in the field of radiotheranostics. And this theranostics is a compound word of therapy and diagnostics. And as you can easily understand that it means we do the diagnostics and the therapy in a single one time. And nowadays, these medical medicine is now emerging to the precision medicine. And the meaning of precision medicine is to pinpoint the patient who will get the better response to the therapy. And as you can see, the imaging has a good promising because we can see whether we can see the lesion, and we can see whether this treatment will respond to this patient. So now, as an old proverb, you can think that seeing is believing, but now we can see and we can treat. So these radiotheranostics have many benefits, because when we use a targeting ligand and radioisotopes, when you use imaging radioisotopes such as gallium or indium, we can actually image and diagnose the patient. And in the same time, when we put in that same ligand a therapeutic radioisotope such as lutetium or yttrium, we can actually treat the patients. So, but this is not a new concept. We have actually been using this concept as a iodine therapy, but now it is emerging to other fields such as a gallium DOTATATE labeled with gallium-68. And as you can see here, we can discriminate whether this patient will have a good response to the treatment or not. And for the patient who has been chosen to have a good therapeutic response, we can then label the DOTATATE with the lutetium and do the targeted therapy. So as a matter of fact, you can see that there is a increasing numbers of the market size regarding the radiotheranostics. From the start, it is actually without the iodine therapy, but with the start of radium, there has been an increase with the FDA approval of lutetium DOTATATE. And now there are many other portfolios that we will encounter in near futures. So actually these theranostics have been, in 10 years ago, actually known as unusual radiological treatment. And in the news for the Steve Jobs, you can see that there was a news that Steve Jobs went to Switzerland and got an unusual radiologic treatment. But now, these unusual treatment has gained the interest of the big pharmaceutical company, which is Novartis, and it has been this Novartis company have bought this AAA company for the lutetium DOTATATE, almost a $4 billion. So there actually is a big interest in this market. And to gain these kind of radiotheranostics agents, there are many hurdles that we should go through. So first, we need a basic research to find a compound, and then for some in vitro and in silico tests are needed to find out the good target. And whenever discovering these target drugs, we have to do the preclinical research. But in each steps, these percentage of the maintaining drugs or candidate drugs will reduce significantly. And after these preclinical research, we can go through to the clinical research. And after phase 1, 2, and 3 trials, we finally can have one approved drug. And lastly, after these approval of drug, these drugs should be in the clinical guideline to be widely used and not be wasted away from the market. And in this kind of path, the importance of the radiopharmaceutical actually stands in the part of the basic research and drug discovery, or in the preclinical research steps. So in this sessions of this workshop, we will deal with what radiopharmaceuticals have their action in drug discovery from bench to clinic. But one also important issue for the radiopharmacist in this drug development and clinical application is the quality control. So now you can see a gallium-68 DOTATATE PET CT. And as you can see from here, this patient has a high uptake in the liver, and also, as you can see here, this patient has a lesion in the pancreas and also a liver metastasis. So this patient actually underwent a lutetium DOTATATE therapy in Malaysia, and this was a Korean patient who had a therapy in Malaysia. And what do you think this next image is? Do you think it's a bone scan, or what is what do you think this scan is? So the answer we will discuss later on the later part using at the quality control part. So today the session will be done from, actually I am quite sorry that there was some change in the course, But so for the first, we will do the theranostic from bench to clinic, which will be done by Professor Yoon-sang Lee, who is the radiopharmaceutical co-organizer, and me, Min-seok Soo. And next, the theranostic clinical application will be done by me. And the third session will be radiopharmaceuticals and developments by Dr. Ji-Yong Park, who is radiopharmacist. And the last will be the theranostic and radiochemistry-based quality control, which will be held by me and Professor Yoon-sang Lee. So thank you, and please join the workshop, and please ask any kind of questions if you have any at any time. Okay, so then we will move on to the next course, which is theranostic from bench to clinic.
Okay. Assalamualaikum. I'm Yoon-sang Lee, the professor of the chemistry team in our department. Today, we can start how can we develop the new radioisotope probe for using the theranostic purpose. So that is based on my experience, and then we can start this concept page. Can you hear me? Okay.
Yes, we can hear you. Yes.
Okay, thank you. So at the year of 1998, I just came our department, and then I saw our facility to synthesize something new. At that time, we have only 3 round-bottomed flask and 1 magnetic bar and 1 rotary evaporator and nothing else. So at that time, I start kinds of N2S derivatives that for technetium labeling for myocardial imaging agent. And then I synthesized several things, and then I gave the sample to our campus in far from here, and then they checked the NMR spectroscopy after 1 month. So I think for structure or information that we synthesized things, we should take NMR spectroscopy. So at that time, we have to spend more time to confirm the chemical structure of new materials. So after 10 years struggle for set up the new facility for chemistry team, so we can get PET-CT scanner and also SPECT scanner for animal study, and also we have an NMR spectroscopy, NMR spectrometer and mass spectrometer also, and fully equipped chemical hold system, and also we have a separate dedicated room for chemical synthesis. That is the basic facility for new drug development. But I think if you want to make a new compound, with others' help, and then you can collaborate with the Department of Chemistry nearby you, and then you can ask to, okay, this compound is a very good potential to develop the Theranostics. Then you can give the chemical structure, and then they can make, and then you can lab by yourself, and then you can start in vitro or animal in vivo studies like that. So normal course of new RI probe development is here. So first of all, you have to select the proper target, and then you can get any kinds of small molecule or peptide or even amino acid and protein like that, and then you can select that which one is best for those target, and then you can select two synthesize method for that chemicals. So, and then after that, because of that compound should have a radioisotope, so you have to do the radiolabeling. And after that, you have to confirm the in vitro experiment and to confirm the efficacy for a cell or protein level. And after that, you can do the animal experiment for a biodistribution or targeting efficiency for with the imaging facility. And then right after those things, you have to pass through the non-clinical study. That is the that needs a huge budget for kinds of theranostic, because of for a diagnostic purpose radiopharmaceutical, you can do the only one injection toxicity study, but for a therapeutic radiopharmaceutical, you have to do the multi-time injection toxicity study. So normally we have to pass through the non-clinical trial stage, then we can submit the investigational new drug application for FDA or Korean FDA to the just like a government body, and then they allowed to do the clinical trial. Then we have to do the clinical phase 1, 2, and 3. Then we can get the whole data, and then we submit the NDA process, new drug approval process. Then we can get the approval from the FDA or Korean FDA, and then we can sell or we can do the clinical use of the new radioisotope probe. So first of all, we have to select the target. So for example, for a brain receptor, we have to do some kinds of dopamine transport or dopamine receptor or anything you can imagine. Then you can select a target. Then you can simply search the Google for a brain D2 receptor and space and antagonist or agonist. You can see the so much. You can see the so bundle of chemicals or peptide. You can see. Then you can print out. Then you can give that chemical structure to the colleague of the chemistry department. Then you can ask to synthesize that precursor or chemical. For the selection of the target, you have to consider that the applicability and also future. As I know that lutetium DOTATATE it has passed more than 20 years, over 20 years. Finally, they can sell that product to the Novartis. Before before the company's success, we know that lutein DOTATATE will be the first first mover to the new era of the nuclear medicine, and then we can select the next one will be the PSMA, and third one will be the melanoma targeting. So I think so many researchers who wants to make a new target, then so many radiochemists devoted into the melanoma target compound. And nowadays, FAP inhibitor will be the next target, but that FAP will be the good target for a diagnostic purpose, but therapeutic purpose, I think specificity will be the problem maybe. So anyway, if you want to make a new Theranostics, then you have to, firstly, you have to select the target. And then you have to select the radionuclide, such as if you want to theranostic purpose, then you have to select the beta and gamma, or beta or any kind of things, any radiation purpose. You have to select the diagnostic or therapeutic purpose. So in case of therapeutic purpose, you can select alpha and beta minus, and diagnostic purpose, you can select the gamma and positron emitter. And also you have to consider that the half-life of radionuclide, because of your target, just like a PSMA targeting molecule, gallium is proper target, proper radionuclide. Because of that, gallium PSMA peptide is after 2 hours, that compound almost gone, passed through the kidney. So which target you select, then that proper radionuclide is there. So simply you can pick the radionuclide from the radionuclide table. And then next part will be the feasibility of chemistry, even including radio complexation or radio labeling. And also finally, we have to consider that the molecular weight or lipophilicity or charge, because of if you especially want to make a new brain receptor imaging agent, and you have to consider that kinds of things also. Here you can see that our small molecule, normally that is the size of several nanometers. Small molecule, from small molecule to the colloidal structure, you can see the sub-micrometer sized molecule. So if you want to make a kinds of sentinel node imaging agent, you can select the colloidal structure for that can pass through the lymphatic pathway like that. And also charge also you have to consider, because of sometimes positively charged or negatively charged particle and negatively charged molecule can pass through the kidney excretion, and also highly positive charged molecule can affect the cell surface. So normally neutral is best one. But sometimes we can give the charge into the molecule because of the some positively charged molecule can pass through the myocardial cell. All negatively charged molecule can pass through the kidney excretion. So if you want to make a myocardial imaging agent, you can select the positively charged molecule. And if you want to make a kidney imaging agent, you have to select the negatively charged molecule. Normally for blood-brain barrier, we have to pass through the blood-brain barrier. You have to select a neutral charged compound. And also Hydro-Philicity that can give the given the non-charged molecule, or hydrophobic one, they possess the alkyl chain or phenyl moiety like that. And then just we can start the synthesis method that any kinds of synthesis method then you can select. Then also even the biological biochemical synthesis also you can select. And as I mentioned that you can ask to synthesize those molecules to the your colleague or nearby department, chemistry department, and then also you can order to synthesize some small chemical company, or even in China, they possess so much of chemical synthesis company, they can make a new compound for you. And then you can label so many or many ways. So here you can see that diagnostic purpose, you can label with the F18 or C11, technetium 99M or gallium-68. For the therapeutic purpose, you can label the iodine-131, rhenium, yttrium, lutetium, and others, or actinium like that. So you can select the adequate radiolabeling chemistry, such as for F18 or C11 or any kinds of metals like that. And also after making that radiolabeled compound, you have to do the purification step. Normally, we can do the HPLC purification for development stage, but for a clinical use, you just simply mixing with that kinds of radioisotope. And then after mixing, no purification, just simply draw that radioactive liquid, and then you can inject into the intravenously. So right before the IND process, you have to do the adequate purification step, purification method, and then you can go into the IND step. And also you have to do the proper QC, quality control method for each radio isotope probe. So you can check the radiolabeling efficiency and also radiochemical purity. And sometimes we need specific activity and also pH or chemical purity. Yeah. So in case of us, almost for a technetium compound, we just use the radiolabeling kit for a technetium labeling. So we don't need any further purification step. And also gallium also like that. F18 or C11 compound, we have to do the HPLC purification, and then we can check the chemical or radiochemical purity of that F18 or C11 labeled compound with the HPLC also. As I know, here you can see that only 3 mechanisms of the radio labeling. You can see the F18C11, that is the SN2 reaction, and other radiometal, that is a coordination chemistry, just simply mixing with the chelator with the metallic radionuclide, and they can make a complex formation. And other one, radioiodine, is a somewhat different mechanism, that is electrophilic substitution reaction, but mainly F18 or other metal chemistry is SN2 and coordination chemistry. So that is very simple, and I think you don't need to struggle for that kinds of chemistry. Just simply you can select which radionuclide is which chelator is good for which kinds of radionuclide, and you can see the simply pick the in the table in the textbook. And then okay, you can get the new radioisotope probe. Then you have to do the in vitro experiment. In our department, before year of 2006 or 7, before getting that PET scanner, we only do the in vitro experiment, and sometimes we can do the ex vivo experiment for biodistribution. But still, we can publish in the good journal. And after getting PET CT scanner, then we can show the whole PET image of our new radioisotope probe. For in vitro experiment, you have to check the stability of the newly developed radioisotope probe in aqueous solution or serum stability. And also sometimes we have to check the charge determination with the electrophoresis or ion exchange column. And you have to check the log P value for repopulity with the octanol distribution study. And then finally, we can do the binding assay with the proper protein or proper cell line for getting information of the inhibitory or dissociation constant of that KD or KI. And also sometimes we can put in the serum or tissue, just like a brain or liver tissue, that radio pharmaceutical get into the tissue and then grind it together, and then we can check the TLC or HPLC. Then we can do expect that what is the metabolism mechanism of that newly developed radioisotope probe. And after finishing that in vitro experiment, we can do the animal experiment, such as ex vivo study, just like biodistribution with the after injection, IV injection of our radiopharmaceutical, and then we can get the whole organs, and then we can check the gamma counting and radioactivity. Then we can show the kinds of graphical information with the biodistribution. And then we can getting information that timely, we can do the timely biodistribution study. Then we can get information of the pharmacokinetics and sometimes pharmacodynamic data. And then we can do the, sometimes we can do the autoradiography, just you can inject into the animal, then you cut the tissue or organ, and then you can impose the X-ray film or some specific material, then you can read out the whole autoradiographic image. And sometimes we can use the animal PET or SPECT or MRI. And then from those data, we can calculate the binding potential for each target organ or each specific area. And all whole data we can say that is effective. So we can expect that efficacy of that newly developed radioisotope probe. And sometimes we have to do the in vivo metabolism study to take urine or fishes, or sometimes we can get the liver tissue. Then we can see what is the metabolite of the from the intact radio pharmaceutical. And then preclinical tests, I think you cannot do by yourself because of that needs a GLP grade preclinical test, such as acute toxicity or any other kinds of series of preclinical study. You have to ask to do that test in the specific institute. So as I mentioned, that we have only lab bench, I think almost 5 years after the 1998. And then we can see serially we can get the NMR and mass, HPLC, and also cyclotron and hot cells like that. And finally, animal PET study, PET CT and Phobicity scanner. So in my opinion, you can start from now. Even you don't have a cyclotron or any kinds of chemical analysis machines, and just you can start, and then you can get kinds of information from newly developed radioisotope probe. Then sooner or later you can get mass or NMR and also TH scanner, and then PET CT scanner in your department or in your country. And then finally, I think you can do everything by yourself. So I think I hope you can do by yourself. That is a very good point to start the new radioisotope probe development. I think that this is the end of my presentation. I think Min-Seok So will continue this session.
So I think we can have some discussion time after this. So if you have any questions to Professor Yoon-Sang Lee, please remember it and please ask him after this short Okay, so what I want to share right now is, so from what Professor Yoon-Sang Lee have told in this time, how these radiopharmaceuticals are discovered and what kind of procedure we need to get to the clinic. And I right now want to share the first clinical trial of the Theranostics, which is the lutetium-177 DOTATATE therapy. And actually this lutetium DOTATATE therapy in Korea is now firstly done in 2020. And as you know, this lutetium therapy actually in worldwide and in the Europe has been started almost from 2001. So actually we are late behind, like 20 years behind these other countries. So I want to share what kind of obstacles we had met to do this clinical trial. So actually in Korea, neuroendocrine tumor is growing from 2000 and it's keep growing right now, but it's still the estimated prevalence of the patient is less than 2,500 patients, and actually it is below the limits of the rare disease definition, which is 20,000. And so there is a trend of increasing patient, but the patient number is still low in Korea, and there is no registry system in Korea. However, not the importance of number, because there are suffering patients. We had to transfer this lutetium treatment to the patients who are suffering. And actually there, besides this lutetium DOTATATE, there are some other options, such as a conventional treatment, such as Sutent, or newly invented targeted therapies, such as everolimus. However, compared to these therapeutics, this radiopharmaceutical, the lutetium DOTATATE, have proven its efficacy for treating patients who had progressed with conventional therapies. So this Lutathera has been approved by the FDA in 2018. But in Korea, it is still, there is no any radiopharmaceutical for the therapeutic proposed that are approved by the Korean FDA. And actually, as you can see, the nuclear medicine volume and our research is quite high compared to other countries. But and so we have international trainees from all over the world. And as I know, some people have come from Indonesia, physicians also have came to Korea. However, our patients, our neuroendocrine patients have went abroad to Malaysia or China, or some have gone even into Europe to get the therapy, because we don't have a clinically approved therapy right now. But we had tried to get this approval from 2010, that we had a project to get a clinical application of first targeted radionuclide therapy. So we prepared for the preclinical studies and toxicity test. However, in 2014, because we joined to the pigs, the radiopharmaceutical also need a GMP standard for the production of these radiopharmaceuticals. So this process halted in 2014. And because of the public demand from the public and suffering patients, and because of some radio pharmacy, which had given an interest to make this drug for the clinical approval, we have restarted this again in 2020, and now we have got a clinical approval for the phase 1 clinical trial. And so there are many hurdles to get into this clinical research, but we are only in the stage of this initial stage of clinical trial, which is clinical phase 1 trial. And the purpose of this clinical phase 1 trial is to check the safety and for the dose finding for the next trials. And to to this clinical trial level, there are many documents and many exams or tests that are needed, such as laboratory tests, and we need a research protocol and also animal test results. So these are some conducts that are needed for the human clinical trial. And there are exploratory studies such as what has been previously introduced by Professor Yoon-Sang Lee, such as pharmacokinetic or pharmacology studies, and also studies that should be done in the GLP facilities, like toxicology tests. So these are documents that are actually needed to the Korean FDA when you apply for a clinical trial preparation. So actually these are in numbers, so but I just wanted to show you that there are so many documents that are needed. But the first main part is about the product itself and the drug substance and the drug product. And secondly, what it is needed is the stability of the product. And the next part is about, and these parts are actually parts regarding to the product characteristics of the product. And the second part is about the safety and efficacy. And actually what we had a huddle was, and as a clinical physician, what we encountered is what we have to deal is more on these safety and efficacy issues. So for the radiopharmaceutical toxicity, there are many toxicity documents that are needed. From the A, it is single dose toxicity test, and B, C, there are other tests. But for the radiopharmaceuticals, actually, there are no genetic or reproductive toxicity which is warranted. And the cause why they are not saying for these kind of documents is because this toxicity test is not for the radiotoxicity, but this toxicity actually refers to the toxicity of the ligand. So it is a pharmacological toxicity of the ligand itself. But in the characteristic of radiopharmaceutical therapy or like diagnostic agent is the characteristic is that we use only a small amount of ligand. So actually this concerns of carcinogenicity or reproductive toxicity of this ligand actually can be ignored in, and so these conducts are not actually warranted for the Korean FDA, and also it is same for the FDA, American FDA. So but other issue is that then there is, so for this toxicity, it actually refers to the toxicity of the ligand. However, the toxicity of the radiopharmaceutical actually is on for the radiotoxicity. And this radiotoxicity cannot be tested like a previous or conventional drugs, which they use like 50 or 100 times more. And so these radiotoxicity can be tested by the biodistribution and dosimetry studies. And actually the Lutathera also have reported this biodistribution and dosimetry studies. But one concern also is that then what kind of form we have to use. So there are a cold form and a hot form of radiolabeled drug. And when the radiolabeled drug has, and these cold form and hot form has the same chemical composition, and when administered at a high dose, the possibility of deaths is not related to the pharmacological characteristics. And so to identify the ligand-related toxicity, we can use a cold pharmaceuticals. And actually, the Lutathera also used using this lutetium-175, which is a cold compound. But there is a concern we have that there is no consensus right now about the alpha particles. Since some alpha particles don't have a cold form, and some radioisotope also don't, if they don't have a cold form, what should we use is our concern. And actually, and also for the radiation toxicity, there is some concerns of late radiation toxicity. And if the patient, like neuroendocrine tumor patients, have a long life expectancy, we have to have a assurance for this late radiation effect. And for these late radiation effect, the FDA actually recommends to do at least twice the maximum planned human dose for radiation tolerance. And so these repeated dose study in the Lutathera has been done in the cold form. However, using the hot form of the lutetium DOTATATE, lutetium-177, they used a double dose of which can be applied to the human dose. So now we can consider that for the radiopharmaceutical, for the therapeutic agent, actually the single dose toxicity study is enough. And also, and it is for the test of toxicity of the ligand, and we have to use a cold form, but there is a concern for like alpha particles which don't have a cold form. And the next is about the radiation toxicity. And what we have to care is that this radiation toxicity has a deterministic effect of the radiation. So if we do the dosimetry study, it can be, we can actually predict the toxicity by calculating the organ doses. So the toxicity assessment for the radiopharmaceutical, like therapeutic radiopharmaceutical, should be done in both for the ligand and also for the radiation toxicity. So there should be a proper combination when you make a regulation. And the hurdle for the Korean to pass through the Korean FDA right now was that there was no actual regulation of these therapeutic radiopharmaceuticals. But now there is we have finally made the regulation in 2019, November. And we always have to participate in this making the regulation, and to have a breakthrough, you must participate. And so what I have some actually questions for whether what kind of regulations are in Indonesia for the radiopharmaceutical or like therapeutic radiopharmaceutical for the clinical approval, like for the FDA, or so. Yeah, that can be, I think, that kind of issues can be discussed. And if you have any questions or comments, please freely ask.
I got a question from the Bang Arianto. So,
yes. Yes, Professor.
Yeah, okay, okay. So you can you asked the binding potential related with the binding potential.
Yeah. In your one year of your slide, Prof, you mentioned that binding potential can must be considered with animal experiment. In Indonesia, we rarely use animal experiment because of the facility. I wonder what kind of binding potential then that you mentioned about and how to measure it, because in Indonesia is rarely to do the animal experiment. Thank you, Prof.
Okay, that kinds of binding potential normally used at the brain receptor study normally. So you can imagine that you can calculate the concentration of radioisotope, and also you have to consider the number of receptor, and then just simply binding potential means the ligand concentration the receptor and ligand binding form per whole ligand concentration. So I think in case of us, we can do the that binding potential can be calculated from the PET study. So because of that PET signal contains the information of the quantitative amount of the how many or how much radioactivity in that focused area. So in my thought, you can do the animal PET or animal sometimes SPECT. Then you can calculate from those data, and then you can get the information about the binding potential. So then I think other method, I don't know, because I'm not an expert of that kinds of study, but I think you can find another method to calculate the binding potential, maybe.
So this is Lee, and
Okay.
I can add to the answer that the Dr. Bang Arianto, he would be interested in what is the meaning of the binding potential in case of tumor model of mice or rats. So as was told by Professor Yunsang Lee, as binding potential is usually used for brain receptor ligand studies. But in case of tumor, so let me just emphasize that the binding potential should represent specific binding in addition to the non-specific binding, which is usually in case of tumor background. Background meaning the blood flow is or blood volume is increased in the tumor. Then the non-specific binding is going to hamper is going to hinder to measure the specific binding. So let me remind you that in case of brain, binding potential is usually the compared with the for example striatum compared with the cerebellum. But in case of tumor, we cannot do that. So sometimes new radiopharmaceutical, which was supposed to have a high binding potential, was later on found to be the blood pool agent like albumin. So many of the monoclonal antibodies presumed to have a high binding potential was later on found to be a blood pool agent. So that happened 20 years ago, 30 years ago, 20 years ago, and 10 years ago. So that might be as I expected, as I speculated, Dr. Bang Bang's the curiosity. And let me just transfer this question to Professor Jae-sung Lee, which is planning the winter meeting, the detailing the tracer kinetic. So your question was really timely. And there has been another question there. Yeah, immunotoxicity.
Yes. Yeah. I have seen the question from medical Dr. Edison, I think, that how to detect immunotoxicity and genotoxicity. For first, actually, I am not very expert to answer this question, but what I want to, actually these tests should be done in the GLP facilities, and for the radiopharmaceutical, for the theranostic radiopharmaceutical, actually these immunotoxicity and this genotoxicity can be waived. So only the toxicity test that FDA or also the KFDA require ask is actually the single dose toxicity. And about the question, Nu Hidayati, thank you for the question. And that the animal experiment to do clinical stages for like estimation of human dose is actually warranted from the FDAs. And what we use is a dosimetry method. And we do the dosimetry test and we do the biodistribution test first in the animal. And then we extrapolate that organ biodistribution to the human phantoms. And then we calculate the doses of the human organs using this animal model. And then how to estimate the dose for the human is based on the maximum threshold of these organs, the organ in risk, such as kidney or bone marrow. So like lutetium DOTATATE that we have, we actually measure the biodistribution of the mouse, and then we extrapolate it to the human phantom, and we calculated the bone marrow dose. And we put the maximum dose to which does not exceed that threshold dose of the bone marrow, which is known to be like 2 gray. So that is, I think, the method that we do and what these governors want us to provide. So if you have any.
Yes, thank you. Thank you for the great answer. And is there any probably like journal material that you can refer to us so we can learn from this in France? Thank you.
Okay. Yes, I can actually, I will share the some regulation files also in the website after this end of this workshop. Thank you. Thank you for the questions. And I think because of the time, I think we will end the first session right now. And I think, we can proceed on to after 10 minutes. Thank you very much.